SpectraMosaic is an exploratory tool developed for the interactive visual analysis of magnetic resonance spectroscopy, or MRS, data. It is the first of its kind to allow comparison of spectral metabolite concentration ratios across multiple dimensions at group and individual levels. MRS is a non-invasive biochemical imaging technique utilized in clinical research for a range of neuropsychiatric diseases and disorders. Current toolsets for MR spectroscopy analysis are severely limited and typically restricted to visualization of spectra for a single voxel for a single patient at a single time, as we see in this figure. In designing SpectraMosaic, we created a taxonomy of visual encodings framed around spectral metabolite concentration ratios used for key attributes of MRS data that fall under four main categories: cohort size, number of spatial voxels, number of acquisition times, and brain state under which spectroscopy was acquired. With our system, we identify 16 potential task use cases highlighted in this figure. For example, Case 9 shows an encoding system for a case comparing spectral metabolite ratios of two patients at a single spatial sample location. We can see our calculations for Case 9 here, where we compare the concentration ratio of metabolite A to metabolite B in a voxel sampled from the same brain region in two different patients. The spectral heat map overview tile C shows the average metabolite concentration ratio of metabolite A for patient 2 to metabolite B for patient 1. Dotted lines leading from the X and Y axes, respectively, indicate the spectral areas used in this ratio calculation. Inside cell C, we perform a different set of calculations related to metabolite A versus B. The average ratio in voxel 1 of metabolite A to B for both patients as a rounded rectangle, while the other two nested discs represent metabolite A versus B for patients 1 and 2, respectively. In designing SpectraMosaic, we chose a two-panel coordinated view. The left panel provides for spectral selection from a structural image reference, while the right serves as the spectral analysis interface in the form of a table matrix. In using SpectraMosaic, users are primarily interested in assessing group versus individual variation of metabolites in ratio form. These comparisons may be for similarity assessment, where subtle differences are important to track, as well as for large difference assessments, such as those seen in tumor studies or in research cases where echo time is varied. This tool was developed in close collaboration over several iterations with experts in MR spectroscopy research. For our final design, we gathered additional feedback from several experts in MRS. Here we show a case study where we look at neuroinflammation in 3 different brain locations compared for 2 different patients. Study datasets are loaded into the application using a drag-and-drop window feature. These data remain linked semantically in the application to ensure continuity between voxel location, its spectral output, and patient-specific information. We can see our patient selector panel on the left. Radial buttons allow display of one patient with associated acquisition data at a time. Here we have selected patient F900 and can see in the image view the 3 voxels acquired for this patient arrayed along this vertical axis. Mousing over each voxel node shows voxel ID and echo time used in the acquisition protocol. Selection of a voxel shows voxel position along the anatomical image. A horizontal axis indicates the number of different time acquisitions performed for this voxel. In this case, we are interested in looking at only one time acquisition. To analyze spectral ratios, we simply click and drag voxels of interest from this left selector panel over to the right. In this case, we are interested in comparing all spatial voxels of one patient to all spatial voxels of the other patient. Once both axes have spectra attached, a heat map matrix is drawn on screen. This drawing updates each time spectra are added to or removed from the panel. Since there are typically 20 metabolites in a spectrum, we have drawn this matrix as a 20 by 20 grid. As we hover over the table cells, we can easily see the segments of metabolite spectra from each axis that are used in ratio calculations, with corresponding area calculations displayed, as well as the metabolites that we can expect to see in this region. Cell size can be adjusted on the fly for targeted analysis of specific metabolites. We use diverging color maps where white indicates a ratio of 1, while red indicates a ratio greater than 1, and blue indicates a ratio smaller than 1. Darker hues represent numerical distance from 1. If the ratio is negative, gold is used to indicate a ratio higher than -1, while green indicates a lower ratio. Each cell can be expanded to show detailed metabolite ratio information for a given cell. For instance, perhaps we would like to inspect individual ratio variation for NAA versus lipids, ml, for this study. On expansion, we can see that as we hover over elements, associated patient spectra are highlighted in fuchsia along the graph axes, in the corresponding table row or rows, which contain useful metadata about the patient voxel sample. A legend at the right lists the key visual elements and maps their shape and color. We can see through similar hues of the color map that both patients in this example are quite similar across all three voxels, although racial tooltips show slight variations. We can see the greatest difference in the prefrontal voxel sample, resulting from a higher ratio for patient F425. Such subtle changes are difficult to understand through visualization of the spectral graph or through statistical measures alone. If we wish to reset the heat map canvas, they can simply click the Reset View button at the bottom right of the interface to re-standardize the spectral grid. To clear the heat map canvas, we click the Clear Data button. SpectraMosaic, when used in clinical research settings, can contribute in the assessment of individual variability, variability across time and space, and to understanding the complexity in the data beyond inference on statistical mean values. This constitutes a novel approach to investigating MRS data.